The heart is comprised of 4 chambers, the left and right atria and the left and right ventricles. These chambers are lined with cardiac muscle fibers which are responsible for generating the force required to pump blood. The left atrium and left ventricle are responsible for pumping newly oxygenated blood from the lungs to the rest of the body. The right atrium and right ventricle are responsible for pumping deoxygenated blood that has just returned from the body back to the lungs to be reoxygenated. These compartments take turns at filling up and ejecting blood in a process called the cardiac cycle. The initiation and timing of each contraction during the cardiac cycle is essential and is fundamentally controlled by electrical activity. While the brain can slow or speed up heart rate via autonomic nervous system pathways, the actual rhythm is set by the heart itself in a region within the right atrium called the SA node. The SA node contains pacemaker cells which generate a signal at about 70 impulses per minute. This electrical signal occurs in the form of action potentials. An action potential is a rapid change in membrane potential, and membrane potential is just the difference in charge between the inside and outside of a plasma membrane. Therefore, membrane and action potentials are a direct result of ion movement. Action potentials can be understood visually by plotting membrane potential on the y-axis and time on the x-axis. When positive charge enters the cell, membrane potential goes up. We call this depolarization. When positive charge leaves the cell, membrane potential goes down. We call this repolarization. The shape of an action potential on this type of graph is distinct to the type of cell, as different cells have different forms of ion movement depending on their function. The action potentials generated by the pacemaker cells within the SA node do not strictly have a resting potential. They first exhibit a gradual depolarization due to the opening of funny channels, which allow for slow intake of sodium ions. This region plays a crucial role in setting the pace required for a healthy heartbeat rhythm, often referred to as the pacemaker potential. This occurs until membrane potential reaches a threshold, which causes funny channels to close and fast voltage-gated calcium channels to open. These allow for fast entry of calcium ions, causing a rapid depolarization, which generates the action potential. These then close and voltage-gated potassium channels open, which allow for slow exit of potassium ions, causing repolarization. Ion pumps restore the ion gradients and the process can repeat. A single action potential in a pacemaker cell can cause ions to move to adjacent cells via gap junctions. This can cause the adjacent cells to generate their own action potential. Hence, action potentials can spread through the heart in this manner. In particular, they first spread from the SA node to the contractile cells of the atria, causing them to contract, and then to the AV node via intranodal pathways. The AV node also contains pacemaker cells and delays signals from the SA node by about 0.1 seconds before they are then passed onto the ventricles. This ensures the atria can contract before the ventricles, which is essential in maintaining the proper cardiac cycle. However, the action potentials exhibited by contractile cells have different characteristics to that of pacemaker cells. Unlike pacemaker cells, they have a resting membrane potential and only exhibit depolarization when activated by a neighbouring cell. Transfer of ions from a neighbouring cell can cause depolarization to the threshold potential. Reaching this threshold causes voltage-gated sodium channels to open, allowing for fast entry of sodium ions. Shortly after, L-type calcium channels open, causing slow entry of calcium ions, allowing both sodium and calcium to contribute to the action potential. When the action potential nears its peak, voltage-gated sodium channels close, slowing down the overall depolarization. Then at the peak, voltage-gated potassium channels open, causing potassium ions to move out slowly. This causes a slight repolarization until potassium exit balances calcium entry. This generates a plateau phase. It is during this phase when the cardiac cell contracts. It achieves contraction via a process called calcium-induced calcium release, which also makes use of L-type calcium channels. Calcium channels close, and net exit of potassium allows the cell to be repolarized. Ion pumps allow the original ion gradients to be restored and the process to be repeated. As suggested by the name, antiarrhythmics are used to treat arrhythmia, which is the broad group of conditions associated with a heartbeat that is irregular, too fast, or too slow. Fibrillation refers to arrhythmias where heartbeat is irregular. Tachyarrhythmias refer to arrhythmias where heartbeat is too fast, which is clinically defined as more than 100 beats per minute. Bradyarrhythmias encompass all arrhythmias where heartbeat is too slow, which is clinically defined as less than 60 beats per minute. Arrhythmias can be supraventricular, which means they originate before the ventricles, either in the SA node, the AV node, or the atria. Arrhythmias may also be ventricular, which means they originate in the ventricles. These type of arrhythmias do not affect the atria. Atrial fibrillation is the most common arrhythmia. Random impulses cause rapid and random twitches of the atria, which makes them ineffective at pumping blood into the ventricles. The ventricles can still pump blood, they just don't receive as much blood with each pump. While not immediately life-threatening, this can cause symptoms such as fatigue, lightheadedness, and chest pain. Atrial flutter is when the atria beat too quickly. Ventricular tachycardias and fibrillations are the most life-threatening arrhythmias, as it renders the ventricles unable to pump blood to the body. Tachycardias may also result from reentrant circuits, which are formed by the creation of two pathways where one loops back on the other. Torsades de Pointes is a special type of arrhythmia that can lead to sudden cardiac death. Antiarrhythmics can slow down the heartbeat either by interfering with the ion channels responsible for cardiac action potentials, or by affecting the sympathetic regulation from the brain. They can be classified into 5 classes depending on their mechanism of function. Class 1 antiarrhythmics block voltage-gated sodium channels in contractile muscle cells. This slows down entrance of sodium ions into the cell, and since sodium ions are responsible for depolarization in these cells, it therefore takes longer for the threshold potential, and thereby contraction, to be reached. Class 1 can be subclassed into 1A, 1B, and 1C. 1A prolongs the duration of action potentials. This makes them effective for slowing down tachycardias, however they pose risk of torsades de Pointes. 1B shortens the action potential duration, and 1C has no effect on action potential duration. Class 2 antiarrhythmics slow the heart down by suppressing sympathetic effects. They do this by binding to beta-1 adrenergic receptors, preventing the influence of catecholamine hormones such as adrenaline, which usually increase heart rate. It particularly slows down conduction at the AV node, making it useful in treating supraventricular tachycardias. One concern is that these drugs may cause AV blocks, which can induce bradycardias. Class 3 antiarrhythmics block voltage-gated potassium channels in contractile cells, slowing down exit of potassium ions. This causes a slower repolarization, in turn causing the effective refractive period to increase. This, however, can increase risk of torsades de Pointes. Class 4 antiarrhythmics block calcium channels. This targets pacemaker cells in the SA and AV nodes, slowing down entrance of calcium, which is responsible for depolarization in these cells. This slows down the depolarization, thereby slowing down the rate at which these nodes generate impulses. However, these calcium channels also play a role in the contraction of cardiac muscle fibers, so should not be used in cases of systolic heart failures. Class 5 antiarrhythmics include all other drugs which reduce heart rate by other or unknown mechanisms. Two notable types include funny channel blockers, as well as agents that decrease calcium levels by other mechanisms. This brings us to our case study of one particular antiarrhythmic drug called digoxin. It is a class 5 antiarrhythmic and functions by downregulating activity of the sodium-potassium pump, an enzyme that plays a crucial role in regulating ion balances between successive action potentials. This causes intracellular sodium levels to increase, which lowers the tendency of new sodium ions to enter via diffusion through the sodium-calcium exchanger. This diffusion usually drives outward movement of calcium. Hence, the drug causes increased calcium retention, which in turn improves contractility. Digoxin also upregulates parasympathetic activity of the vagus nerve, which causes the SA and AV nodes to produce signals at a slower rate. Hence, digoxin contributes to both a stronger and slower paced contraction of the heart. Digoxin may be taken orally or injected intravenously. Digoxin has been proven to have therapeutic benefits for various arrhythmias, most notably for atrial fibrillation, as well as heart failure. However, digoxin has no proven benefit on mortality rate and has a narrow therapeutic window due to its ability to induce adverse side effects when taken in high doses. Side effects may range from nausea to induced arrhythmias. For patients prescribed with digoxin, oral dosage should be between 0.125 to 0.25 milligrams per day, and plasma concentration should be maintained to be 0.5 to 0.9 nanograms per milliliter.